back students to Genetic Engineering and Genomics Module 3. In this module, we'll be learning DNA fingerprinting. So let's start with the definition. What is DNA fingerprinting? DNA fingerprinting is a technique used by scientists to distinguish among individuals of the same species using what as samples, my dear students? DNA as samples. You know why? Because in our DNA only there are those unique sequences called as VNTR, variable number of tandem repeats, and that is why it acts as what? Key factor for this technique called as DNA fingerprinting. This technique was discovered in the year 1984 by a very famous scientist called as Sir Alec Jeffreys. Let's start with this technique. So in this technique, what we'll be doing? We'll be taking DNA or we'll be isolating DNA. So DNA isolation is done from either blood, semen, hair root or other samples from where we'll get the DNA. So for example, if I isolate DNA from blood, here's the DNA isolated. Now if the amount of DNA is less what has been isolated, what I'll do, I'll amplify using a technology called as PCR, what you have learned in the chapter 3, biotechnology, process and application. So polymerase chain reaction will help me to increase the number of copies of DNA. The next step, as the DNA is very long, I need to fragment them into small pieces. So let's start with the second step, DNA fragmentation. The DNA is fragmented using enzymes called as restriction endonucleases, and this will result into fragments, and I'll get variable size of fragment, and hence this technique is also very called as restriction fragment length polymorphism, that is RFLP. Now after getting these fragments, I need to separate these fragments, and I'll separate using a technique called as agarose gel electrophoresis, a very famous technique for separation of molecules. So let me just show you a diagram of an electrophoretic apparatus in which there is an agarose gel. It's a jelly like substance which will help the DNA to move, and there is punched well in this gel, and there's a solution. In the well, I'll load my fragmented DNA. Now the name suggests electrophoresis, means the movement of DNA is on the basis of electric current. In the next picture, you can see that we'll apply a voltage current through which the DNA, as it is negative charge, will move from the negative electrode to the positive electrode, and hence the DNA fragments move from the negative to positive side. Do you want to see that how do they move? On what basis? Being on the electric current, but how they'll move? They move depending on their length. Shorter the fragment, faster it can run, and longer the fragment, it will be on the top. And this is how you can see in the picture, it results in the formation of bands. Now moving to the third step. The next step is the bands are then blotted on a nylon membrane. But before that, this gel is placed in an alkaline solution so that the double-stranded DNA gets converted to single-stranded or opened up into single-stranded DNA. And the single-stranded DNA is then blotted on a nylon membrane. So single-stranded DNA gets embedded onto the nylon membrane. And this technique is called as Southern blotting, as we are using DNA as sample. Now after doing this, the next step is the hybridization part, where I'll be looking particularly for those sequences which is making us unique, that is VNTR, and that is why I'll flood this membrane by radioactive probes which are complementary to VNTR sequences, but excess of probes are washed off using water. Correct? The next thing, now what has happened, that the single-stranded DNA and where those VNTRs are present, the radioactive probes have been complemented and excess has been washed off. So the next step or the last step of this technique is photography, where I'll place a X-ray film onto it, and due to radioactive probes, the photographic image will come onto X-ray. And let's see how does this X-ray film looks. This X-ray film looks particularly over here. You can see in the picture where Sir Alex Jeffrey is holding this X-ray film, and this is called as autoradiograph. So this is how this technique is carried out, and the final output is in the form of X-ray film called as autoradiograph, where it is particularly showing you only those VNTR sequences or those unique sequences. From India also, Dr. Lalji Singh developed a probe called as BKM, that is Banded Crate Mini Satellite Probe for DNA fingerprinting. He obtained this from a Y chromosome of a female banded crate snake. A very good picture of a female banded crate snake. Now let's just see applications of DNA fingerprinting. DNA fingerprintings help me for parental disputes. It also helped me for catching criminals or also in forensic studies. And the major application is for genomics. We'll be soon studying this genomic in the next module. Till then, my dear students, I complete DNA fingerprinting. In the next module, I'll see you with the genomics and a very important thing, Human Genome Project. Till then, thank you, students.